Welcome to our channel Bio by Gulab Sir for Wisdom Coaching Center. As a part of series of lectures, the topic is polyembryony. That means many embryos. Generally, each seed have only one embryo that we know. Polyembryony generally it is a characteristic feature of gymnosperm. It is very often found in characteristics feature of all gymnosperms. So remember this as it is a characteristic feature of gymnosperm, but it is very often observed in mango and citrus family plant. Citrus, that is orange, sweet lemon and so on. So what is it? How it occur? Here it is clearly shown in diagram. This area is ovule. Here this is embryo already developed, zygote, and written as an embryo, that embryo which developed from zygote. But at the same time, some of the nucellar cell, these are the nucellar cells. These nucellar cells divide and redivide. So some of the nucellar not nucleus, nucellar cells which are deployed, no doubt, around developing or around the developing embryo. Divide and redivide mitotically. Divide and redivide mitotically. And produce embryos. Produce many embryos. This phenomenon is known as polyembryony. I hope you did understand. Here this is clear diagram that these are the nucellar cells around this developing embryo. They divide and redivide and produce a mass of cells which is functionally similar, structurally and functionally similar to original embryo which developed from zygote, and hence that many embryos are produced in one seed. If we crush a citrus seed, that you will find more than one embryo, and that phenomenon also has to be studied in practical. So you will come to know if you see. This is exactly what the definition, that some of the nucellar cells around the developing embryo, this was a cells. Later they divide and redivide to produce mass of cells which are functionally similar to that embryo is developed from zygote. The difference is that this embryo is developed, it is by the fertilization from male and female gamete, and this embryo is developed from the diploid cells of that nucellus belong to that one plant. Because to understand the difference between these two embryos, this embryo is formed by the fertilization between male and female gamete, that what by fertilization, and this embryo is formed by the nucellar cell, diploid cell, which belong to only one plant, and therefore there may not be variation. Here it is variation which has two sets of gene from two different plant, but here two sets of gene belong to the same plant. This is the major difference between these two embryos which are produced here. We are concerned with this more than one embryo developed by the nucellar cell, and that is called polyembryony. And yes, it is observed in mango and citrus plant seeds. I hope you did understand this phenomenon of polyembryony. Second part of this video is apomixis. Now, what is apomixis? Apomixis is a process of production of seed without fertilization. Now, how it happen? It happen sometimes naturally also in the plant belong to Asteraceae family. Apomixis is a phenomenon of production of seed without fertilization, and that seeds are known as apomictic seed. So definition can be written: it is a process of production of embryo or seeds in embryo in seed, production of without fertilization. Is known as apomixis, and the seeds which are developed, seeds that are developed without fertilization or produced without fertilization are known as apomictic seed. Now, why these are important? This is possible naturally as well as artificially. Naturally, apomictic seed are developed or produced in plants belong to some plants, not all plants. In some plants belong to family Asteraceae. This Asteraceae is also known as compositae, because question can be asked in either way by having name Asteraceae or compositae, as well as in some of the species of grasses. So please remember these two Asteraceae and some of the species of grasses where these apomictic seeds are naturally developed. But why this is important artificially to the farmers? When we produce a seed, desirable seed with two desirable characters, just now we have first video we learned artificial hybridization, that bringing two desirable characters together in a plant, new variety with that two desirable character. Now that two desirable character together, maintenance of these two desirable character together is a challenge. Why? Because when that plant developed with that two desirable character grow and have that flower, in the process of gametogenesis, these two character may segregate. You know very well what is segregation. Is the separation of that two genes which present on two chromosomes. During gametogenesis is a law of segregation, and by that character which brought together may get separated. Now to maintain these two characters together is a challenge, and therefore apomictic seeds are costly to the farmer. So to maintain that two desirable character together has to have development of that seed without fertilization, because that nucellar cell or cells other than zygote, here it is shown aposperi, that is what development of here these is aposphere, that initial diploid cell around the megaspore mother cell. Megaspore mother cell which supposed to undergo meiosis, but it won't allow. That is arrested by using artificially by using acetylcholine. Acetylcholine won't allow to form the spindle fiber, and therefore there will not be a meiosis, and that megaspore mother cell will remain as it is, or the cell just neighbor to the megaspore mother cell, that what diploid N to N is continue to be a zygote, and that zygote undergo division and redivision to develop the embryo, and that embryo which developed will have that both the character together, because meiosis was not allowed. Mitosis occur, and with that mitosis, here it is clearly written apomictic reproduction, aposphere, that is what formation of zygote and embryo without spore formation, that means without formation of egg, without fertilization. That diploid cell is as it is allowed to undergo mitosis and produce the embryo with that diploid condition. Here the egg cell is developed without fertilization, and that is diploid. This egg cell as it is become embryo without fertilization, without having gametogenesis. Therefore, it is known as aposphere. Here this is natural sexual reproduction, how that two character which bring together that may get separated from each other, and thereby there may be chances of production of separation of that characteristic. So as we have knowledge of meiosis, that there is in meiosis 1, when metaphase completed and anaphase begins, there is a separation of homologous pair of chromosome. So in that homologous pair of chromosome may carry that desirable genes present on two different chromosomes in two different gametes, and thereby that character may get segregated if meiosis occur. Therefore, to prevent the sexual reproduction and have the apomictic reproduction, that is production of zygote, development of diploid egg cell without gametogenesis, that is called apospore. Apospore is the process of formation of spore, that is egg, diploid egg cell without fertilization, and that diploid egg allowed to develop that embryo as it is with that two desirable character is known as apomixis. You can refer this diagram in our video as and when it will be uploaded, so you can expand it, or you can go on net and search. You will find this is given on net. Here this is natural process. When meiosis occur, there is a separation of that characteristic enabling to produce that two embryo with two separated character, and our very aim to bring that two character will be defeated to have that both the character together. These are the two processes that is given here. Mitotic diplospore, that means diploid cell is continue to be diploid. That egg cell which produce megaspore mother cell act as a egg cell without undergoing meiosis, having both the character together and developing that embryo with both the desirable character is apomixis. I hope you did understand, because you are well aware about meiosis and gametogenesis, how that character can be segregated, that you already learn in genetics or may learn in genetics principle of inheritance. As in mitosis there is no separation of character, so that megaspore mother cell is allowed to undergo mitosis and not meiosis by using acetylcholine. That means spindle fiber formation is arrested, and the two sets of genes with two desirable character present on two separate chromosomes are continued to maintain inside that seed to have that new plants, develop that seeds and plants with that desirable character. So to maintain this apomixis, or to maintain the two sets of character, desirable character in one seed, required a trained hands and to have that trained skills, and therefore these seeds with two desirable characters are costly to the farmer, because they are assured to have that two desirable character in that seed without any fail. Yes, once again, what is apomixis is nothing but production of seed without fertilization to maintain the desirable character. It can happen naturally as well as artificially. Naturally, it's very often observed in asteraceae. Some plants belong to asteraceae and grasses, as well as artificially it can be done, and therefore it has importance in farming to produce seeds with our desirable characters. I hope you did understand what is polyembryony and apomixis. Here, this what explanation for polyembryony. Once again, you can refer this one. This is what definition for polyembryony with example that polyembryony is a characteristic feature of gymnosperm. In gymnosperm, there are more than one archegonium that get fertilized by different pollen grain so as to produce more than one embryo, and therefore gymnosperm has polyembryony in common for all the plants. But in angiosperm, since there is only one zygote on one embryo, therefore they do not have polyembryony. But in some of the plants belong to angiosperm, that is mango and citrus, they belong to angiosperm. Even though in them that more than one embryos are absorbed, which are produced the way it is explained over here. This is ovule, this is nucellus, this was embryo developed from zygote, that is zygotic embryo, and these are the embryos developed from nucellus, therefore nucellar embryo. The difference between these two is this zygotic embryo is developed by fusion of two gametes, that one male gamete from one plant and another male gamete from another plant, or maybe belong to same plant, depend if self pollination. But this is what these embryos develop from nucellus. Definitely they have both the sets of chromosome of the same plants. Therefore, they are assured to have that all the characteristics belong to that same plant, and this is the major difference between these two embryos. But we are here to understand what is polyembryony. Therefore, the embryo, more than one embryos developed from nucellar cell is polyembryony, and observed in mango and citrus family plant. What is apomixis? That is what development of seed without fertilization is called apomixis, and the phenomenon is known as apomixis, and the seeds which produce are known as apomictic seed. This process is natural, that is sexual reproduction. How that desirable character may get separate, that is shown here, and here how that desirable character is maintained, because here that cell, megaspore mother cell, won't allow to undergo meiosis because of artificial effort, use of acetylcholecystine, arrest the spindle fiber formation. Therefore, there is no separation of that chromosome which has two desirable character. They remain together, and mitotically that zygote develop into embryo, which develop into the plant to produce the seed with both the desirable character, and therefore that seed is known as apomictic seed, and the phenomenon is known as apomixis. I hope you did understand what are polyembryony and apomixis. Even if you are in doubt, you please ask in comment section or call on mobile number 9898431081. Also, I appeal you to subscribe our channel as well as like it, and press bell icon to receive new videos as and when they are uploaded. Thank you.